Thanks, Mark. Tough act to follow with Ernie Small giving us the real overview of the botany of the cannabis plant. I mean, as you know, it's a very complex material. We are, faced with the idea that it, can be reduced to this sort of single molecule drug, a white pill or an injectable substance. Cannabis is just unbelievably complicated, both chemically, botanically. As Ernie was saying, we still really don't, know how it evolved and how humans first started to use it. My work has sort of tried to shed light on some of that, and I'm going to review a little bit of it today. It's in the context of, I guess the idea for this particular session is that we're going to actually have a panel that Caroline McCallum and I will both be presenting on, and I think is Daniel Schechter is also going to come up. And that is to deal specifically with this question of strains. And Ernie mentioned that in his talk. If you talk to patients, if you go to a dispensary in Vancouver, if you go on to Aurora or Canopy or LP website, you see a number of cannabis strains listed by name. Sometimes their chemical attributes are provided as well. And patients receiving medical cannabis are faced with making decisions about what sort of strain they should have, what's going to work best for them. And I think there's a lot of confusion out there about what strains mean and how we should sort of deal with it in the medical context. And so the Mark asked us to sort of address those questions, I think, this morning. Is it? No, that's yeah, I took it. I brought one. Thanks. Just to disclose one conflict to make it clear, I am the CEO, founder, and a major shareholder of a cannabis testing company and a breeding company in Vancouver called Anandia Labs. So just to make that clear but I'm not representing. We don't make cannabis products, for example. We're not an LP. We work for many of the ACMPR LPs out there. Potential bias. I haven't been involved in putting together the sessions or planning the speakers or what have you. I think I've got two of the same slide here. So what is a strain? I pulled up this definition from the web. And that is that a strain is defined as a breed, stock, or variety of an animal or plant developed by breeding. It's a relatively ad hoc term. It's an informal designation. It has no real status in the horticultural world or sort of formal botanical world, in the sense that typically we refer to plants like roses or canola or what have you as a cultivar, a registered cultivated variety. Strain has arisen in cannabis mainly because we didn't have a formal horticultural system for cannabis. Growers were, planting seeds, producing a wonderful plant that they probably smoked and felt that it had some attributes that were worth preserving. So they either continued to propagate that plant vegetatively or maybe crossed it with another plant. And in this way, hundreds and now thousands and probably tens of thousands worldwide of different cannabis types are called strains. But it really, we talk about flu strains, for example. It's not, not a term that's only used in cannabis, but in the plant world, we don't talk about strains of apples or strains of corn. And so it's really a product of a world of sort of an unregulated system, and the fact that humans typically want to classify things, they want to name things, and so we've applied this strain name, and we're kind of stuck with it now, at least for the time being. There's been a proposal. I know the sort of title of the session talks about chemovars, so chemical varieties, and there's some support for using chemovars to describe what are now called strains, but certainly strains are what patients are going to be talking to you about in their offices or learning about when they go to LP websites. And as I said, cannabis is unique in the sense that, other crops that humans use, apple varieties, tomatoes I show here, rice, potatoes, what have you, or larger acreage crops like wheat and corn, have registered varieties that have known attributes, and the names are fixed to the plant with those attributes. So you have a golden delicious apple. It is what it is. It is a golden delicious. In cannabis, we have something that's much sort of stranger and more chaotic. This is just a sort of a shot. I think I went onto a website, Leafly, that's a sort of U.S.-based sort of strain review website. I just pulled up a whole bunch of names and did this sort of, I think it's based on their numbers of reviews. So Girl Scout cookies, OG Kush, White Widow. These are the sort of names that you often see on licensed producer websites. Certainly if you go into a dispensary in Vancouver, these will be on the sort of the whiteboard menu in the corner. And many LPs rename these strains, but they often continue to connect the current name to the White Widow or what have you. So we have that strain naming convention. We also have, as Ernie mentioned, this very sort of generally recognized but fairly unclear distinction between indica and sativa types. So Ernie mentioned the sort of evolution of cannabis as it goes back to northern India and Afghanistan and other places as being the sort of early development of the idea that there's separate populations or gene pools that might have, this indica type, this sort of short, squat plant with very broad leaves, and the sativa type, the sort of taller, lankier plant, more open, narrower leaves or leaflets. And so that's the world of sort of plant evolution and taxonomic treatment of the plant. But it's also now sort of infused the world of cannabis consumers in that sense that indica plants are believed to be a separate subtype that has not just those morphological features, but has a distinct sedative or body stone type effect, this sort of couch lock, which I have to say, just to put it out there, is not due to higher amounts of CBD. In the sense that, I don't think it's incorrect that at some point indica type plants might have had more CBD, or certainly CBD was present. But if you go into, if you get a cannabis strain from a licensed producer that's labeled indica and compare it with a strain that's labeled sativa using chemical analysis, they might both contain 18% THC by dry weight and 0 or 0.2% or whatever CBD, so negligible amounts of CBD. So it's not that, I just wanted to put this to rest. Indica is not sedating because it has higher amounts of CBD. That's not the case. Sativa, on the other hand, this taller, lankier plant has, what in cannabis lore would have thought to have stimulating or more stimulating or cerebral effects, less of the sort of sedation. So those are the sort of two types. My lab actually has done a lot of work on cannabis genomics and DNA sequencing of cannabis. And what we did at one point a few years ago, this was a paper, this is a figure from a paper published in 2015, was we took a large number of samples of different cannabis types, so hemp types that growers had labeled as indica, types that growers had labeled as sativa, and we did genotyping by sequencing, so this is a sort of a scan sequencing method. And really ask the question, genetically, what do the sequences tell us? And what we found was that hemp was very different. Hemp was a distinct group, separate from the drug types. But there was some suggestion within the groupings generated by the DNA sequencing that sativa and indica could be the sort of different groupings, and it may be a sort of lingering effect from, as Ernie talked about, the sort of original gene pools that have now been hybridized by breeders. So I guess there was a suggestion of differences genetically between these groups, but what we found, in fact, was that the ability of growers or seed suppliers to accurately say if they had indica or sativa type plants was very low. In fact, there was like an R squared there, you can see 0.36, so a moderate correlation. And that was where we said, the plant type was labeled, or was given a so-called reported ancestry. That would be what the grower said it was. Was it 100% indica, 100% sativa, 50-50? Sometimes you see these really crazy numbers like 65% sativa and 35% indica, which is just impossible to have. There's really no evidence for those sort of fine-tuning of the difference of hybridization. But we took the reported ancestry and we compared that to what the genotyping had told us in terms of the genetic ancestry, and they were poorly correlated. So when a grower says this is a sativa type plant, most of the time they don't know at all, and they're labeling that very inaccurately. We also found that strain names are often confused in the sense that people will want, say, a particular strain, Blue Dream or White Widow or what have you, and because there's been very little formal horticultural registration and controls and regulations of those plants, they're often mislabeled. So you can get a plant that is supposed to be White Widow and it's actually Purple Kush, or you could have a plant that's sometimes it's the same plant where different names are applied to it. So there's a lot of mixing up in that world. That's not to say it's all, we don't want to throw the baby out with the bathwater, and that, people are trying to classify plants here. They're trying to put a name to something, trying to sort out the diversity that's present in cannabis. So in some cases, that's working, but a lot of the times there's a lot of confusion there. This is my own version of the sort of entourage slide. The mixture is the medicine. What I've tried to get across here is like the use of strain names is one method to sort out the diversity in cannabis, but probably more accurately is the ability to look at the different chemistry of the plant, because the chemistry is really what's driving the effect, whether it's the psychoactivity that's desired for recreational purposes, or the medical properties. And so cannabis, of course, many people have said this in today and yesterday, is that it's a very complex mixture of chemicals. There's over 100 cannabinoids known. THC and CBD are the two main players. There's hundreds of terpenes present, so those organoleptic molecules that Ernie referred to, limonene, for example, myrcene. They all are mixed together in those little trichome gland heads that we saw pictures of earlier, and together they elicit the effects of the plant: pain relief, appetite, stimulation, etc. So it's that mixture is the medicine, I think, is important to note. And just digging in a little bit, these are the main cannabinoids that we find. So THC and CBD are the two main ones. But other compounds like cannabichromene, CBC, cannabigerol, CBG, and their acidic forms, so they're found as acids in the plant and decarboxylated to their neutral forms by smoking, are also present. So these would be the sort of main six that we would worry about or try to quantify, I think, when we think about cannabis. We also have lots of terpenes. So there's, as I mentioned, there's probably 40,000 terpenes found in the plant kingdom. They're also called terpenoids in some cases. Cannabis probably has a couple hundred of them, and they vary across different cannabis types. But the two sort of main classes of terpenes in cannabis are the monoterpenes, which have 10 carbon structures, things like myrcene and pinene, and sesquiterpenes, which have 15 carbon structures, so beta-caryophyllene is a major player. And when we do analysis, this is the kind of thing that my lab in Vancouver generates. These are in the upper panel. It's an HPLC trace of cannabinoids. So this is liquid chromatography, in this case using UV detection. And this would be a plant that's a mix of CBD and THC, and it's labeled CBDA because it's the acid that's found in the plant. But you also see CBGA and cannabichromene, CBCA as well. And then below that same plant, we've used a different technique, gas chromatography, to look at the terpene components. And you can see there's monoterpenes like myrcene, which is the sort of dominant monoterpene, and sesquiterpenes like beta-caryophyllene. So it really is a really complex mixture. This is just a snapshot of one particular sample. So when we look at THC and CBD levels in cannabis strains, I think this is where we really have to put our focus, not so much on figuring out, is it an indica strain or a sativa strain, but thinking about the chemicals that drive that plant and having a good way of knowing what they are and what levels they are. So back in 2015, I think I started giving this CCIC talk in 2014, but in 2015, there was only 15 licensed producers. Now there's about 95 of them, I think. And they were called MMPR, Marijuana for Medical Purposes Regulations. And there was about 200 strains available. And I went on the web and I pulled out the THC and CBD levels that were reported by the LPs on their on the strains that they were available. And I graphed them here. You can see THC by dry weight on the x-axis and CBD by dry weight on the y-axis. And what we get is this clustering of a large number of strains right along the x-axis. Those are the high THC, low CBD strains. The average at the time, back in 2015, was 17.5% THC. Then there was this other group that was the mix, those sort of one-to-one, that sort of off in the middle there. And then there was a few strains, only very few, that were high CBD, low THC. Since that time, we've seen, a large, there's been an explosion of product types that are available. My lab does a lot of analysis. I got someone to pull some data on samples that we've run recently. So 968 samples that were analyzed from LP cannabis and patient cannabis. And what we saw was, so the high THC, so-called chemotype 1, were dominating. 796 of those 968 samples were high THC, and the average was still, 3 years later, 17.5% THC. So that's the normal sort of ACMPR product that's sort of available generally to patients. We can see the spread there is from less than 10% THC acid by dry weight up to almost 30%, but we're not seeing levels higher than 30%. Typically around 27 or 28% is the maximum level of THC that seems to be available in the ACMPR. When I say patient samples, I mean that under the ACMPR currently, patients who grow cannabis for themselves are able to send it for testing. They're not compelled to do that, but they can do that. So that number also captures samples that are not commercially available through LPs, but the majority of these samples would be through LPs. Then we also see a much larger availability of high CBD or so-called chemotype 3 plants. So these were 89 of those 968 samples, and the average in those plants in that chemotype 3 was about 12% CBDA. So generally, plants that contain CBDA are less potent than the THC-containing plants. Not always, because we can, there's some high 20s or 20-ish%, which is different than even a few years ago. There's been a sort of increase in CBD levels. And then there's this mixed type, the so-called chemotype 2 or mixed THC CBD type, which I don't get into the genetics here, but basically the plant enzyme-wise can have an enzyme that just makes THC, or it can have an enzyme that just makes CBD, or it can have a copy of each, and it can make both of them. And then it's sort of a tug of war between the two metabolites. And so we have, in this case, the chemotype 2 is this mix. It's kind of a, in a way, it's like a combination of drug type and hemp type. But in this case, we see an average of about 10% CBDA and about 7 or 6.8% THCA in 83 samples. And I think this is where some of the interesting medical opportunity lies for LPs and for doctors, in the sense that higher amounts of CBD and and lower potency THC is I think a useful place to be, certainly with naive users, elderly patients. I'm not a doctor, I have a PhD, but I've been through enough talks with Mark Ware and hearing physician advice that I think this chemotype 2 area is where medical opportunity lies, but also in the context of the sort of heated discussion yesterday with the CMA representative, like, will these types of plants survive in a purely recreational context? I'm not sure. I mean, there's a lot. Obviously, recreational is mainly interested in high THC amounts, and these chemotype 2 plants might and their next step, the high CBD only plants, might be sort of ignored in that context. So I think, there's some plant genetic reasons for having two systems in place. So it just, where things are going in 2018, there's more and more strains available. Higher CBD levels, as I mentioned, they're creeping up. There's also higher THC levels, and there's more of those chemotype 2 strains available. Digging in a little bit on where we are in terms of cannabis pharmacology that drives these strain differences. I mean, initially, if we talked about medical cannabis 10 years ago maybe, or 15 years ago, there would be just a focus on THC. THC being the principal, bioactive cannabinoid responsible for the analgesic properties, or most of them, for example. So, I think Health Canada labeling, if we had medical cannabis products available 20 years ago, would probably just have THC on them. And now we realize that this very abundant cannabinoid CBD, or CBD acid, is present and mediates some of the properties, or contributes to some properties. And now we have labeling for cannabis that's the number 2 on this list, THC and CBD. But then there's also the idea that the pharmacology is not just THC and CBD, it's the other compounds present, the minor cannabinoids and the terpenes. And this slide I actually borrowed from Corey Harris's talk last year. I thought it did a good job by saying that, the first hypothesis is sort of being discarded as we learn more about things like strain-specific effects. And and I think we're also going to be discarding the second hypothesis, which is that it's just THC and CBD. And we're kind of moving towards this idea that what really drives cannabis pharmacology, not cannabinoid pharmacology, but cannabis pharmacology, is the mixture is the medicine. And I'll quickly go and turn this over to Caroline fairly soon. Ernie also referred to the entourage effect. This is not something new, and in fact, Ethan Russo didn't invent it, but he's been a big popularizer of this idea that when patients have said, I've tried 2 different strains, and one has this, far better impact on my condition than this other one, and yet when I look at the THC levels, they're exactly the same. And the question is, what is producing those strain differences? And so one idea is it's the minor cannabinoids, the things other than THC and CBD. Another is that there's terpenes that are co-occurring with the cannabinoids that give the cannabis smell, but also have some pharmacology themselves. And Ethan has popularized this idea that they're forming a metabolic entourage where THC and CBD would probably be the king and queen, and then you have this courtiers and other people hanging on, and they form the entourage. I emphasize that this is a hypothesis that needs to be tested, which this is not textbook stuff yet. It actually makes a lot of sense, but it's also built on a lot of circumstantial evidence. So there's very little strain-by-strain comparisons in the scientific literature. I pulled out this paper, and again, sort of acknowledge Corey Harris for the slides that we co-presented last year. A paper in 2014 in Holland, I think, looked at 102 adults that were receiving 3 different strains. So a 19% THC, so a chemotype 1 strain contained very little CBD, a 12% THC, also low in CBD, and an approximately 1-to-1 low THC, low CBD strain. And it looked at things, psychometric scores, such as alertness, anxiety, irritability, etc. And the results that they found no strain differences in terms of consumed dose. This was done in I think a Dutch pharmacy context, Mark. Do you know the Dutch pharmacy? There were no strain-specific effects found on therapeutic satisfaction, so that was, did it work for those patients consuming it? But they did find differences in subjective effects. And some of the ones that were found to be quite different were appetite stimulation, so they found higher amounts of appetite stimulation in the two high CBD or high THC strains and less in the white bars is the low THC, higher CBD strain. And they also found lower amounts of anxiety in that 1-to-1 strain, which sort of makes sense, I think, when you're thinking about a pharmacy population or a population of consumers that are getting their material from a pharmacy who might not be consuming large amounts of and having a tolerance for THC. If THC is provoking anxiety, and Matt talked about this in adults, that a lot of the time people are sort of putting aside cannabis use post-adolescence as anxiety and paranoia. Having lower amounts of THC and higher amounts of CBD probably makes sense there. So there was some differences there. So to summarize and conclude things before I turn this over to Caroline to talk about how patients are dealing with the strain issue, and I think we're going to have a fairly sort of interactive discussion around her case studies. Again, strains are not recognized by horticultural authorities, so this is kind of an ad hoc cannabis-specific naming convention. There's hundreds of strains available in the ACMPR currently. They're genetically distinct, but that genetics are kind of confused, I guess. The indica and sativa classifications are dubious, and I don't mean to say that there's not differences, and there probably were original gene pools that led to the kind of cannabis types we have now, but just the ability of whether it's an LP or a dispensary or a grower or a seed company to say this is 100% indica or sativa is not really there. These strains vary a lot by the metabolite diversity, so the cannabinoid and terpene chemistry is very distinct, and that's probably driving a lot of these reported or purported strain-specific effects. But there's still very little scientific evidence, trials or other comparisons to support these differences. And I guess we always have to conclude our talks by saying more research is needed, and that's certainly the case here. Thanks very much.